Hello and welcome. Today we are going to discuss Fungi, the Thallus organizations. Myself, Hamid Al-Mali. I am doing my MSc Agriculture Plant Pathology at Scottish University. So the topic is Fungi. How the Thallus is organized in Fungi. We will discuss these things. First of all, contents. What we will discuss? We will discuss Thallus and its classification, mycelium and its classification. We will discuss septa, and in septa we will discuss the special case of Doripore septum. After that we will discuss fungal tissues, and then modification of the hyphal structures. There are following 4 modifications usually, which is rhizomorph, aprosorium, ostium, and traps. So let us discuss Thallus and its classification. What is Thallus? Thallus is a vegetative body or the fungal body. It is usually hyaline, or it may be colored if the pigments are present in the cell wall. It is usually achlorophyllous, and just like the plants, it's not divided. Its body is not divided into roots and stem and the branches and all that. Whole body of the fungus is called a thallus. And now we will discuss the classification based on number of cells. Based on number of cells, thallus is divided into unicellular thallus and filamentous thallus. Unicellular thallus is the one which is made up of single cell, while the filamentous thallus is the one which is made of filamentous structure, or the filamentous structure which is branched and is multicellular. Unicellular thallus containing fungi include Chytridium and Synchytrium, while filamentous thallus include Alternaria and Pythium. Now, the classification of thallus based on reproductive structures. Based on reproductive structures, thallus can be divided into eukarpic and holokarpic thallus. In this picture, you can see eukarpic. Eukarpic thallus is the one in which the thallus divides into vegetative part as well as into reproductive part. Here in this picture you can see monocentric and polycentric thallus. Monocentric thallus is the one in which single reproductive structure or sporangium is formed, whereas in case of polycentric, many reproductive structures are formed. Monocentric can be divided into endobiotic thallus and epibiotic thallus. Endobiotic is the one in which the reproductive structure or sporangia is formed inside the cell, while epibiotic is the one in which mycelium, which is reproductive structure or the sporangium, is formed on the cell. Holokarpic is the one in which whole of the thallus gets converted into reproductive structure. After vegetative part of the life, the whole of the vegetative body gets converted into this reproductive structure and is known as holokarpic thallus. Example of eukarpic thallus is Pythium, and holokarpic thallus is seen in Synchytrium. Now coming to mycelium. Mycelium is network or aggregation of hyphae. Mycelium can be hyaline, that's colorless, or it can be colored also if the pigments are present on the cell wall. Based on septa, mycelium is divided into septate and sinostic. Septate fungi is the one in which the cells are separated by transverse walls called a septa, while as in case of sinostic or non-septate thallus is not divided into septas, or the thallus is not separated by septas. Only in case when the reproductive structures are formed, this septa is formed that gets separated from rest of the thallus. Examples of septate mycelium include ascomycetes, basidiomycetes, and deuteromycetes. And examples of sinostic mycelium include oomycetes and zygomycetes. Now, based on nucleus, mycelium can be divided into homokaryotic and heterokaryotic. Homokaryotic is the one in which 2 nucleus of the same genetic origin are present, while as in case of heterokaryotic mycelium, two different genetic nucleus having different genetics are present in the same cells. Coming to septa. Septa is the transverse wall which separates the two cells. Septa is can be further divided into two: primary septa and adventitious septa. Primary septa is the one in which cells, after the nuclear division, when the nucleus divides, along with that the septa is formed. That is, septa is laid down between the two daughter nuclei. While as in case of adventitious septa, this kind of nuclear division or the nuclear division is not followed. This septa formation is not followed after the nuclear division. You can see the image of the septa here. Now coming to electron microscope studies of septum. Septum can be divided into simple porite or dolipore septum. This is the simple porite septum and this is dolipore septum. Simple porite septum is found in ascomycetes, whereas dolipore septum is found in basidiomycetes. This dolipore septum is important, so we will discuss the structure of dolipore septum. These are the two images of dolipore septum. In the left side you can see the red colored septal wall or septal palate. While moving through the cell wall to the center, you can see that in the center, when you approach to the center, the septal palate or septal wall flares out and a barrel shaped structure is formed, and this barrel shaped structure is called a dolipore septum. On either side of this barrel shaped structure or dolipore septum, two structures, you can see these structures. On the right side you can see these black structures. These are called parenthyosomes. These parenthyosomes allow only some of the materials and stop some of the materials, or the communication between the cell is controlled by this parenthosome, and it is differently named by different scientists. Coming to the fungal tissues. Fungal tissue is actually a pseudo tissue and is known as planktonchyma. Fungal tissue can be divided into prosenchyma and pseudoparenchyma. Prosenchyma is the one in which these mycelial threads are loosely arranged, and these can be seen, or it can be somewhat differentiated. But pseudoparenchyma are the fungal tissues which are more or less likely to that of the parenchyma. It is very difficult to differentiate between parenchyma and pseudoparenchyma of the tissue, parenchyma of the higher plants and pseudoparenchyma of this fungi. So this is called pseudoparenchyma. Coming to the modification of hyphal structures. Hypha is modified in the fungi into rhizomorph, aprosorium, hostorium and traps. We'll discuss each of them. First of all, rhizomorph. Rhizomorph is derived from the Greek word rhizom means root and morph means shape. The growing tip of the rhizomorph resembles that of the root tip of the hair plants. That's why it's called a rhizomorph. The most studied rhizomorph structure is that of the honey mushrooms, Armillaria melli, and this is the image of roots of or the rhizomorph of this Armillaria melli or honey fungi. These rhizomorph, what they do? They pull out the nutrients while getting the nutrients from different areas and pull them, and fungus uses them. Coming to the aprosorium. Aprosorium word is derived from Latin word aprimere, means to press against. It helps the germ tube or the hypha to attach to the surface of the host or substrate. You can clearly see how the spore has germinated, and you can see the germ tube, and after germ tube, you can see this abrosorium. Abrosorium is actually used to attach the spore. It holds the spore upon the tissue. And after this attachment, penetration peg enters into the between the cells. After that, hausturia. Hausturia is also derived from the this Latin word hauster, means drinker. This is also a special structure or outgrowth of the somatic hyphae, and is used to absorb the nutrients from the this cell. It's important to see and know that this when the hausturia enters into the cell, it ruptures the cell wall, but it does not rupture the plasma membrane. Although it ramifies or branches extensively in the cell, but it does not rupture the plasma membrane. There is a thick covering of this hausturia, which is known as zone of apposition, which is formed between the wall of hausturia and the plasma membrane of the cell. The vesicles of the host plant attached to the transfer the food through this cytoplasm of the host and enter this zone of apposition, and then enter the haustural wall, and hausturia absorbs this and takes the nutrients from the cell to the fungus and supplies this to the rest of the thallus. Coming to another modification of the fungal tissue, that is traps. This is the modification of hyphae in predaceous fungi for capture of small animals, protozoa, rotifers, and nematodes. Some of these trap fungi belong to zygomycetes, and most of these belong to fungi imperfectae or deuteromycetes. You can clearly see how a nematode has been trapped by the fungus here also. And we will discuss that there are two types of traps usually. One is called adhesive trap and another is called non-adhesive or mechanical trap. This adhesive trap, there are following types. Some adhesive traps are three-dimensional, forming three-dimensional adhesive networks, and some are forming two-dimensional adhesive networks. Some form adhesive branches, some form adhesive stalk or knob, some form non-constructing ring, some form sessile adhesive knob. And this, in case of this non-adhesive mechanical traps, three-celled constructing ring is formed. And how this three-celled structure or the adhesive or mechanical trap captures this nematode or the prey, its prey. These three-celled structure, when some the prey, or like in case of in here, you can see this nematode when it enters into this ring. Immediately after failing, the fungus swells up to 3 times of the normal cell, the normal size of the cell. It swells 3 times of that, and the nematode gets trapped. And slowly, when the nematode is not getting the food, this enters this hausturia into the nematode and takes the nutrients from the nematode. There's a short video in which it is shown that how this spore germinates and forms the penetration peg and forms the abrosorium and penetration peg and this hausturia.
This spore must now find the nutrients it requires to grow. Sending out the germ tube, it seeks an entry point into the plant. A newly formed penetration tube breaks inside the stem, and the fungus extends further into the plant. Once inside the stem, another structure, called the haustorium, is used to penetrate inside one of the plant's cells. The fungus can now take nutrients from within the plant.
Thank you.